Yeah, I'd say so.
This is
Yeah.
the beautiful organism called
Yeah.
Anna Bender. She has long, thick brown hair. How does she get this? Well, let's take a look inside her cell to find out. Now, this is the microscopic cell in Anna's hair where the building of the protein takes place. And we're going to zoom even farther into the nucleus where transcription begins. During transcription, mRNA is made from the DNA. However, they differ greatly in characteristics. DNA has a double-stranded structure, while RNA is single-stranded. DNA has 4 nitrogenous bases, adenine, guanine, cytosine, and thymine. RNA has the same 4 nitrogenous bases except uracil instead of thymine. They both include a deoxyribose sugar and a phosphate group. mRNA's polymer is a nucleic acid and its monomer is a nucleotide. The first stage in transcription is initiation. The promoter sequence indicates the beginning of the gene. That is where the RNA polymerase attaches to the DNA strand. Then elongation takes place. In elongation, the RNA polymerase reads the DNA strand from 3' to 5' and builds corresponding RNA strand from free-floating RNA nucleotides. The RNA is then being built from 5' to 3'. The RNA polymerase eventually reaches the last stage of transcription, the termination sequence. This part of the strand tells the transcription to stop, and the RNA strand breaks off and moves to the mRNA processing stage. In mRNA processing, at the 3' end of the RNA strand, a poly A tail is added, which is a sequence of 150 nucleotides. Then, at 5' end of the RNA strand, a cap is added. This will allow the ribosome to recognize the mRNA sequence and bind to the mRNA when translation begins. The RNA strand contains introns and exons. Exons code for a protein, but introns do not code for a protein. Therefore, introns must be removed from the RNA strand to produce a mature messenger RNA strand. This process is called intron splicing. Once mRNA goes through this process, it is able to leave the nucleus through a nuclear pore and into the cytoplasm, where translation takes place. This process is where the protein is made from mRNA. Translation starts when the 2 ribosomal subunits are put together, the smaller one and the larger one, which are composed of rRNA.
The ribosome units attach to the mRNA at the start codon. A codon is 3 nucleotides grouped together. tRNA has an anticodon and an amino acid. It attaches to the complementary codon on the mRNA strand inside of the ribosome or translation complex. When the tRNAs bond to the mRNA, the amino acid from both are joined together to form a peptide bond. As the RNA strand continues to move through the ribosome, more tRNA is matched with the strand to form more bonds between the amino acids being released, forming a polypeptide. Eventually, the mRNA will reach a stop codon, ending translation.
Amino acids break off into a protein.
This protein that was just made is what causes Anna's hair to be brown. But when the mRNA strand is altered, called a mutation, it changes the protein that is made. A nucleotide in the mRNA strand is either changed, taken out, or inserted in. When a nucleotide substitutes for a different nucleotide, this is called a point mutation. A nucleotide being inserted into the mRNA strand or taken out is called frameshift mutation. Either way, these mutations usually, but not always, changes the amino acid sequence. For example, in this mRNA strand, the mutation that occurs is a substitution mutation. The CGA codon, the guanine nucleotide, being substituted out for an adenine nucleotide, creating the codon CAA. This will change the sequence of the amino acid, forming a polypeptide. This means this protein does not code for the trait of brown hair anymore. Since it has been mutated into a different amino acid sequence, the protein does not have the same function. Therefore, it is not one that contributes to Anna having brown hair.